It's a special night, and we're really pleased to see such a wonderful turnout, and to see so many friends. Unfortunately, there are a lot of people who want to be here, who couldn't be here, and we'll have more to say about them in a few minutes. This is the 50th anniversary of Herp Group. Marvalee and I started Herp Group in our in our basement, basically.
No, our first house, upstairs in the living room.
Oh yes, that's right. It was upstairs in the living room in our first house.
Then we moved up to the basement.
On Fairlawn Drive, up in the Berkeley Hills. When we arrived from Chicago in the fall of 1969, Berkeley was still on the quarter system. And we didn't start until just about the 1st of October. And as a result, we didn't start Herp Group until about 2 weeks in. So this is almost exactly the 50th year of Herp Group. It was modeled on something that we did at Chicago, where Marvalee and I were at different universities, and we had a group that would meet at our house near the University of Chicago campus periodically. Not as regularly, when we arrived at Berkeley, we discovered there were a lot of herpetologists around who didn't even know each other, and some of them did and weren't interacting, and there were no such things as lab group meetings in those days. As a result, people didn't have much to bring them together. And so we thought we'd have a Herp group because everybody was problem-oriented in their own ways. That was not the issue. The issue was how could we get people who were working on the same animals from many different perspectives to come together. And the first year, we would meet weekly. We did meet weekly for, oh, I suppose, 10 years? About 10 years. And in our home. Some people from that era are here tonight. I'm very pleased to say. George Gorman.
I was here then.
You did make it, I remember it well.
I mean, in '69?
George was a postdoc here. He got his PhD at Harvard. He was a Miller postdoc. And then he had another year postdoc, I think, with Alan Wilson.
Yeah.
And then he became a professor at UCLA, and now he's retired and back in Berkeley. Paul Licht, comparative physiologist in our department, was among the very first people to attend Herp Group. And he's still here at Berkeley, having been professor and chair, dean, a fallen herpetologist. He fell a long ways. But he's back in the fold, and we're happy to have him.
I never left it in my heart.
I know. In your heart. You know, Al Bennett and Rudy Berkelham. Al was a Miller Fellow as well. Postdoc in Paul's lab. He went on to a very distinguished career. He and Rudy were both at the University of California at Irvine. He became all the things that Paul did, including a dean of college biology, college life sciences, I guess. And now is retired as well. We both retired. And back tonight, and we're so grateful to you for being here.
It was a wonderful time.
It was. We have only good memories.
There are a few other people I'd like to introduce, and I do this at a great risk because I'll forget people, and I'm counting on Marvalee to help me.
Well, you have a list. That's handy.
I do.
No, it's a different list.
Oh.
Todd Jackman and Andres Galazo, who are both graduate students here. Andres is the only student who is co-sponsored by me in Marvalee.
Poor guy.
Still the only one, no one ever came after me.
A very courageous man.
And Andres is now at Caltech. Todd is now at Villanova. And we've got people here who received instruction from Todd at Villanova and are back for their PhDs here. And there she is. Then Cango Ballet, who was here on sabbatical, and I think you were here at least 2 different times. And
I wasn't here in 1969.
no, you weren't. You attended Herp group. He's mainly an ichthyological paleontologist, but also a herpetologist. I'm glad to have you here, and your wife Kate.
Thank
Welcome.
you.
Happy to have you both here. Our dear colleague Jim Patton.
What else can you say?
He thinks of himself as a risen herpetologist. He's a mammologist.
He raised the food for gopher snakes.
Jim is on so many of our students' doctoral committees, and so has been a part of this scene, an honorary part of this scene for a long time. We're happy to have you here tonight. And he's here tonight because of Tom, in part, because he was a member of Tom's doctoral committee, and Kent Lightfoot, professor of anthropology, who is Tom's major professor, is here. So delighted to have you here.
I'm taking notes tonight.
Lu Zeng is back with us, another member from the past. And then we have Dan Portick. and Raina Bell. Raina was here as an undergraduate. Now she's back in the area, and we're so delighted to have you here too. Bob Hansen, been a long time associate and now formally a formal member of the Museum of Vertebrate Zoology. We're delighted to have you here. And Bob is the editor of Herpetological Review and very well known to herpetologists around the world. Dan Edwards, another person who has an appointment here in the museum, is here from UC Merced. Dan, delighted to have you here today. So I'm sure I've forgotten many, but those are people who I especially wanted to know, and we welcome you all. Now, a few items of business. We have a full program for Herp group. A week from tonight is our next Herp group. And then we have another one in 2 weeks, or 2 weeks after that. And that's the one at Cal Academy. Cal Academy is 2 weeks after that. So we have 3 coming up, including one at Cal Academy. And we'll deal with the issues of transportation and things like that to Cal Academy later. All right, so now it's on to a moment in the history of herpetology. And I'm going to turn not to Ted Pappenfuss, who's away and can't be here tonight, unfortunately, but to Marvalee, co-organizer of Herp group for all these years and still always here loyally. So here you've got a moment for herpetology.
Well, I actually have the pleasure of giving you several small moments in history. In this case, the history of Herp group. When we sent out the announcement, we encouraged people to tell us, if they wished, what they remembered about Herp group, and some people did. and some of them are hysterical. But first, I'd like to say that several people responded and, of course, weren't able to come, but they said they're with us tonight in spirit. These include Richard Wassersug and Eric Lombard. I'll tell you more about Eric in a moment. Steve Arnold, who's apparently leaving for Peru toward the end of the week. Jim Hankin, Javier Rodriguez, Brad Schaefer, John Losos, Aaron Bauer, who noted that his first herp group was 37 years ago. Ray Huey, who's backpacking in Ethiopia. Kayana, Sean Kupta, Craig Moritz, Lori Klosterman, Rachel Mueller, Inigo Martinez Solano, Paul Elias, Dave Blackburn, Robert Kaplan, Barry Sinervo, Matthias Stock, Chris Schneider, Martin and Julie Feeder, both sent some memories, just wait, Gloria Wurst, and a number of others. But let me just give you a few of the memories that people presented. First, I'll tell you Eric Lombard's. He claimed the title of founding graduate student with Wassersug. He and Richard came to Berkeley with Dave and me as Dave's graduate students. They both got their degrees from Chicago. And Eric made the point that he carried the herp group concept with him and transferred it to Chicago in another guise. He formed a group that became the Evolutionary Morphology Group at Chicago in the mid-1970s, and that group continues to this day, and it's run by the Committee on Evolutionary Biology at Chicago. Jim Hankin, who finished in the late 1970s, had 3 memories to offer. The first one, Raina, cover your ears. One was when someone was working with Al Leviton at Cal Academy, and he presented the results of his morphometric study of some species. He wanted to discuss the results of a linear regression analysis of 2 variables. It was in the early days of computers, well before canned packages like R, or even SPSS, and Al Leviton, who was regarded as the CalACAD's computer expert, had written a program to perform and display linear regression analysis, and the student had used it for his data. The time came for him to show his results, and he clicked the projector to show the data slide. There was the regression, of course, but instead of a straight line, there was an extended curvy line that went through every point, wandering its way across from the origin at the lower left to its final destination at the upper right. It took about 10 minutes for the laughter to subside. That's what Jim thought of first about herp group. His second favorite memory is the herp group that coincided with Halloween that fall. And we decided to dress up as our research animals. My weak attempt to come as a Thorius involved a large sombrero and other Mexican, quote, quote, garments, but the hands-down winner was Ted Paffenfuss, who came as a bipiece. He carved up an empty Clorox bottle, plastic, to fashion as his head, and he used oversized asbestos gloves to be the hands, and he fastened of bipes, and he fastened them at his neck. They were used in the electrophoresis lab for picking up hot beakers. And Jim says that was truly inspiring. Another extremely memorable moment that night was Lee Van Valen, who was visiting Virginia Majorana, a student of Dave's, singing his dinosaur extinction song solo and a cappella. Great fun. Brad Schaefer got his bachelor's here at Berkeley, but it was a regular attendee of herp group as well. And he says he began going in 1975, And he certainly remembers, he says vividly, individual meetings by Ray Huey, Bob Drews, Dick Heighton, among others, many others. Nancy Staub says, I have many fond memories of Herp Group. That's where I first met David Hillis. That's all she says about meeting David Hillis. I first presented my work. I was very nervous, but had fun later talking about Anides flaviputatus behavior, taking bets about which salamander would prevail in agonistic encounters. Sean Kupta, more recent graduate of the group, said, I miss Herp Group and MVZ very much. I also miss California, and I still dream about Encetina and about human levels of humidity. He's at Ohio now. And outstanding, affordable burritos. Lori Klosterman, who was my student in the mid-70s, presented several favorite recollections of Herp Group. The young son joining in, she says, parentheses, Lucky Tom. The special feeling, fortunate privilege, of having, and fun of having a gathering for real science, but on a casual basis with professors and guests. And her third memory is Ted Pappenfuss, just Tedness. David Darda, who completed his PhD in 1988, has memories of Herp Group, hoping my 1967 Impala station wagon would make it up the hill to your house. The gathering downstairs in front of the pool table, true. Dave tapping on something to call things to order, as he attempted tonight. My first Herp Group was a mere 39 years ago. Fond memories. Julie Feeder has a great one. Thanks so much, but for me the meeting that stands out most clearly is when Sam Sweet ate toad legs that I had carefully skinned and sauteed with butter and garlic in your kitchen. When Ted, Pappenfuss of course, put us onto the possibility of hybridization of Boreas and Punctatus at Darwin Falls, and Martin and I went out there and came back with some putative hybrids, Sam was skeptical, going so far as to say, if that's a hybrid, I'll eat it. Well, it was, and he did. And the last one that I'll offer is from Martin Feeder, who completed his PhD with Paul in 1977. Herp Group taught me two things. And this one, I'm afraid some of you youngsters won't even have a context for. When the speaker shows up an hour late with 4 full carousels of slides, beware. The second one is how to feign pleasure when sampling a disgusting wine, learned from Ted Pappenfuss. There are some memories of Herp Group and moments in history.
Well, we wanted to have an appropriate speaker, and
Couldn't find one.
we wanted somebody who was there at the beginning, who had witnessed years of Herp Group, actually. And we really thought we could do no better than to ask our son Tom. Tom gave his first paper at Herp Group. It was about finding Diadophis. We used to have very informal Herp groups, and people talked about notes from the field, gave observations of what they'd been doing, what's going on, just, quick notes. He saw this, and when he found a Diadophis on his way home from school, he decided he wanted to give a report. He was 7. He later gave some other reports as well, including one that he reminded me tonight is still unpublished. And he has the data.
Still have data, yeah.
So,
Armenides vocalization.
so Tom got his PhD here at Berkeley in the Department of Anthropology, working with, under the direction of Kent Lightfoot, in zooarchaeology. And he's been at UCLA now for a number of years. He's now director of the Zooarchaeological Research Lab. And tonight he's going to tell us about his work in California. So we're happy to have you here, Tom.
Thanks for the kind introduction. Well, I can proudly say I attended my first herp group 50 years ago. Because I was there. I may have been asleep. But I was at least...
Many of us were.
And through the years, I got to see some amazing things related to herp group at my parents' house. Lee Van Valen stomping around with his ode to dinosaurs. Quite impressive. Talking to Stephen Jay Gould about baseball in our living room was also a memory for me. And there are plenty of others. And you know, Ted's cashew wine from Ghana was horrible. And we all remember that. Those of us that can see still. But what I'd like to do today is first thank all of you for coming out. And thanks for having me up to address you. It's a real pleasure and an honor for me. And it brings back a whole lot of memories. I mean, I grew up here in Berkeley. I grew up in this museum. I remember running around in the museum before it was remodeled, getting under everybody's toes, you know, asking questions all the time. Being out in the field was great. I remember specifically being able to capture lots of salamanders with my nimble little fingers and high energy, to the point of where I would orbit Sam Sweet as he would pull over a big log or a rock and then dive in and swoop on all the specimens, often enough that he started yelling at me. Things like, "Get away from my log!" "With my rock!" And I'd be like, you know, little kid, and Sam's big. And I was like, come on. But yeah, I have lots of fond memories. Growing up here in the museum, going to high school around here. All my degrees are from Berkeley. And I moved on to UCLA. And what I do at UCLA is an outgrowth of what I studied with Kent Lightfoot and Cango Ballet over the years too. I focus on zooarchaeology. You might ask, what is zooarchaeology? Zooarchaeology, as I see it, is the study of all animal remains, under a large umbrella, all animal remains that come from archaeological sites. And there's an important caveat here that bears on what I want to talk about today. Not necessarily all the bones you find at an archaeological site are culturally significant in archaeological terms as being sources of foodstuffs. Some are important in ritual, medicine, healing perhaps, but it's hard to pin down those things. Most zooarchaeologists focus on meat contributions, hunting practices, resource depression, change through time, as maybe technology relates to the animals that show up at site. And I do all that. It's extremely interesting to me. And I like to include ethnographic information, information concerning identity, culture change in many of my studies. And my dissertation was at Fort Ross, looking at primarily mammal remains that we excavated over several years there. And Jim and Kent signed my dissertation, so I guess it was okay.
Not too late.
I keep hearing that. And one of the, so I've really, since I'm born and raised Californian, I bleed blue and gold. I cannot help but be interested in things Californian. Vertebrate, zoology, ecology, California ecosystems are all really, really important to me, and I find them fascinating. I have branched out. I work in the neotropics also. I'm a research associate at STRI in Panama, and I have a long-term field project at Sitio Drago and Isla Colón in Bocas del Toro, Panama. I'll leave you with this image. It's archaeology on the beach in the Caribbean. Right. And that's another talk. But what I want to focus on today are results from zooarchaeological analysis that I've conducted at UCLA over the years, from archaeological contract analyses or research-based programs. Basically, people send me collections of bone of all types, and I've boned up a lot of my osteology, and I feel confident about identifying fish, amphibians, reptiles, birds, and mammals in California and some of the neotropics too. Not to brag. Sometimes it drives me crazy. But one of the things that really interests me is the remains that are not culturally important. They're still there. And I see those as, along with the culturally relevant or important, economically important specimens in these assemblages, I see those as representing the Holocene fossil record, with a distinct difference. In archaeological sites, the bones are primarily mitigated, brought to the site, collected, added to the site by humans as part of their subsistence practices, their hunting practices. But these sites also tend to capture local vertebrates that are on the landscape as well, and they get included in the fossil record. So one drum I've been beating a lot lately in terms of conservation biology and biological research is that zooarchaeological assemblages represent the Holocene fossil record, which hasn't been paid much attention to yet, although it's becoming more and more important as we start thinking about the Anthropocene, climate change, and actions that humans have had on past environments through the Holocene. As an outgrowth of that archaeological analysis, I also began to pick up paleontological analyses, contracts as well, because I was interested and I felt I had the expertise, and I wanted to take a look at Pleistocene, late Pleistocene microfaunas in California as well, and compare those to Holocene microfaunas. Again, looking for distributional changes, paleoclimatic reconstruction, and so on. And so what I want to talk about today are a combination of results from archaeological analysis and late Pleistocene analyses seen through a view of the kind of information that these analyses can provide for biological and conservation research. First of all, I'd like to dedicate this talk to my very recently deceased friend, mentor, and colleague for over 30 years, Dwight Simon. He was one of these guys that knew everything about California archaeology, and I think he would have really enjoyed this talk. So, it's for you, Dwight. I'm going to talk about 7 sites throughout California. 5 in Southern California. Let's see if the laser pointer works. And don't look at the laser pointer. I learned that. Not just now. There's a site at Huntington Beach, which I have published on in 2009, but I got a bunch more material from. It's late Pleistocene. There's another late Pleistocene site here in Chino Hills I want to talk about. Then archaeological site at Malibu, SBA 53, right in downtown Santa Barbara. And then the youngest site, which is a real Holocene fossil site from the Avenal Creek Bridge on State Route 41. I also want to talk about a couple of Northern California sites, the famous Emeryville Shell Mound, Alameda 309, and the Castroville Mammoth site, which is really quite interesting. These are all quite interesting. And I could go on and on and on forever, but I'm going to try to keep calm and be respectful of your time and concentration. I'd first like to talk about the Huntington Beach site, or LACM 7679. Basically, it's right here, literally right on the beach. This is the Santa Ana River as it's canalized now, but the Santa Ana River contributed to the development of these estuaries through the Pleistocene, off and on, as the river meandered back and forth on the coastal plain. Zooming in, we're talking about the Pacific City block. When I was there and got to see it as it was being excavated, this entire block was, there are some oil pumpers still there. It had been sold, and there were rolling kind of back beach dunes there, and it looked like a beachfront to me, and everyone was there. There was an archaeological site here. There were a few burials. They've been reburied, but there are also several organic lenses, dark soil in these sand dunes that were pretty compacted and looked promising for paleontology. And my friend, colleague, and mentor, who also recently passed away this spring, Mark Roeder, sent me the remains from Pacific City and a bunch of other sites that I'll mention for analysis, because he was great with fish. He's an excellent fish analyst, and he knew I could handle things with legs. And just for a bit more perspective, this is the Huntington Beach Pier. This is Pacific City. That's where this site lies. And these are Santa Ana Mountains in the background. So, the assemblage from this site dates greater than 48,000 years ago. It's Rancho Lybrand in period. Extinct fauna associated with it include Mammoths, bison, and equus. There is a paper out, Wake and Roeder 2009, in Quaternary Research that reports the first phase of analysis. And as I said, these samples were from dark organic soil lenses in stabilized sandy dunes, in quote. And recent mitigation has added another 1,200 specimens to the list. So now I've been able to identify 44 genera, 31 species representing 32 families in this late Pleistocene microfauna. There are 3 freshwater fish represented, 3 salamanders, 3 anurans, 4 lizards, 7 snakes, 10 birds, and 16 mammals. This is an extraordinarily rich and diverse Pleistocene microfaunal assemblage. There are few like it in Southern California. As far as I know, in terms of Rancho Lybrand time phase, the only more diverse assemblage is found at the tar pits, and that's way more diverse. The freshwater fish found in these soils include the arroyo chub, Gila orchidai, 3-spined stickleback, which is Euryhaline, really, and Cottus asper. fairly common Southern California coastal creek fishes. The salamanders encountered right at the beach in these dunes include Batrachoceps, probably major, Anides lugubris, and the first quaternary fossil record for Encetina schultzi. And I was fiddling around with interest in distributions and what do these locations mean. And here's something from AmphibiaWeb and Berkeley Mapper showing in blue dots actual collected specimens in MVZ and I guess elsewhere that are on record. The orange is expected range for Anides lugubris. And some of these records are a little anomalous, but the red triangle represents Huntington Beach, where you could put Anides on the beach. And I, if I were more creative, I could have come up with an animated picture of Anides in board shorts on a surfboard or something like that, but that's for later. So, and I'm going to show a couple other maps like this to kind of emphasize what I'm trying to say, that as part of the Pleistocene and Holocene fossil record, these kinds of sites can add distributional data to our current understanding of the ranges of these species. There are 3 anurans. I've been told it's okay to say Bufo boreas, Rana draytoni, and Pseudacris Sierri hypochondriaca, I guess. But they're all present in pretty good numbers, especially Pseudacris. So here is a dense data slide showing the 2009 assemblage and then 2 additional assemblages that were sent to me, totaling 1,459 herps represented. Sticklebacks and Orochubs are fairly well represented. The most common salamander is Anides lugubris. There's also Encetina and Batrachoceps, toads, rana, lots and lots of Pseudacris, and then also a good representation of various lizards, including Aniella, Bulgaria, the usual suspects, and a nice representation of snakes as well, including small ones, big ones, and some rattlesnake, and also some pond turtle as well, which, surprise, surprise, leads me to infer that 50,000 years ago, this area, this deposit was a riparian zone in oak, chaparral, savanna, somewhere. Of course, the shoreline was probably a few miles further to the west, and would have come up and down, because 50,000 years ago was the last interglacial period before ours. So the site may exist because the sea level never rose past that and eroded it away. I don't know, I'm not a geologist or a geomorphologist, I can't say. But I found the herpetological diversity stunning. There are also birds. Clapper rails, avocets, ducks, quail, spinus, and an asio. I mean, these are all represented, again, hinting at riparian zones in oak savanna, chaparral things, and the birds include clapper rails and avocets, so waterfowl are the most common, and then sparrows and other such creatures as well. And I have immense respect for David Stedman, because he does this, that's what he does. He's an avian paleontologist, Pleistocene, Holocene, and trying to figure out sparrow distal ulna is hard, but there are similarities that you can pull apart, and I don't know how he does it, but this is as far as I can get. Now, one other interesting thing about Pacific City's fauna is that there are a lot of mammals, including these guys. Outside of cave sites and some Maya temple sites that were, might be bat roosts, I haven't identified bats from any archaeological sites. And as far as I know, they're fairly rare in late Pleistocene paleontological sites too. And I identified 2 Parastrellus mandibles and what looks like a Tadarida molar. Another really interesting thing about this site is the small rodents. Right, there's Paragnathus, there's Dipodomes, probably Agilis, and then there are a bunch of chipmunk teeth. And here is where I could come up with another nice anime of Alvin in board shorts on the beach with his brothers, because they're chipmunks at Huntington Beach. I do not know which species they represent, but they're all molars, so I should come here and look, maybe.
You may be able to sort by size, but that's all.
Yeah, they're small.
Chipmunk species,
Yeah.
they do differ in size, so
Yeah.
they may be able to sort that.
Yeah, and they're rooted molars, so so there's chipmunks and salamanders at the beach, Huntington Beach 50,000 years ago. And here's a list of all the mammals. Pretty diverse mammal assemblage. And for those of you who are interested in these data, I'm happy to show or share later. I know they can be a bit tedious. So I'd like to move on to another late Pleistocene site that dates to about 40,000 years ago. It's called the RP2 site in Chino Hills. It was a storm sewer mitigation project where they did paleo monitoring. They found large extinct fauna, bison and equus, and decided to do soil sampling. And that's where these microfaunas come from, are soil samples, bulk samples, that are washed through fine mesh screens down to at least 0.5 millimeters. And from the RP2 site, I was able to identify 15 genera, 13 species that represent 12 families, including 1 freshwater fish, 2 salamanders, 5 anurans, 3 lizards, and 5 snakes. So these are the Chino Hills, and the sewer improvements were along here, along 57. So it's in a canyon, so probably a riparian zone. This is what Chino Hills State Park looks like on the very best day you can ever be at Chino Hills State Park. And some of the suspects that we found include Bufo boreas, Rana draytoni, Tarica turosa, Thamnophis probably hamidae, but maybe not, and actinemes, or western pond turtles. When I mapped the locality and the collected specimens versus expected range for Tarica turosa, this is the map that came out. And the thing I find intriguing is that my dad and I checked out a couple of these records here in the basin. They seem anomalous, maybe mismapped, because the directions and localities didn't jive. And there's this gap between, Santa Ana Mountains, Tarica, and San Gabriel Mountains, Tarica, and Santa Monica Mountains, Tarica, and Chino Hills specimens sit right in there. I don't know. There are no records of Tarica from Chino Hills. Don't know when the last record could be from, but at least we can place them there in their expected range. at 40,000 years ago. Again, this is a species list. There are 864 herps and a couple fish. And I include fish because I like fish. I like to analyze fish, and my friend, mentor, and colleague Cango Ballet is here, and he's a fish person, not merely a fish paleontologist, but a morphologist, and also an archaeologist, because he's identified lots and lots of fish assemblages from archaeological sites too. The thing that stands out here is the 220 Tarica specimens. I haven't really identified Tarica from any other archaeological sites or paleontological sites in the state other than this one, and they dominate. And again, this assemblage looks to me like something not unexpected from the Chino Hills, maybe oak savanna, open grassland, maybe chaparral complex along bordering a riparian zone. Now I'd like to move to another late Pleistocene microfaunal site that has two components. And this is often commonly referred to as the Castroville Mammoth site, which is actually right here. Microfauna site is right up here, a little higher up. And for those of you who know, the giant artichoke is right here. It's a very important place. Memories. We would go there when I was a kid to Castroville and get deep fried artichoke hearts at the giant artichoke. And we did that last, this spring? So, with my daughter. And the discovery of the Castroville mammoth was due to happenstance and hinged on natural science education of the farm owner's son who was supervising grading operations and noticed that one of the graders clanked against something and scraped up a mammoth tooth, right? And he knew what it was, and he knew its importance, and convinced his dad to allow him to call my friend and colleague Mark Hilkema, district ranger for California State Parks in the area, to come down and take a look at it. And Mark came out, and it was a big to-do in 2008. A bunch of people from Foothill College at Santa Cruz and other places converged to excavate the mammoth. And the mammoth is really quite incredible. I mean, it's probably a washed-in mammoth. Of course, all the archaeologists were hoping for a Clovis point sticking out of the mammoth's ear, but that didn't happen. But there were other extinct Pleistocene megafauna, and it's a good sample of a mammoth. And the matrix that it was in is fine silt. And the preservation is so good that they were able to collect mammoth hair. Right? So, and the things you can do now with hair are amazing. And so the samples of mammoth hair are still available, locked away in a cold safe somewhere in Mark's basement, probably. So I heard about this at the SAAs when they were in Sacramento, and said, of course, me being me, I was like, who cares about a mammoth? Are there tiny bones? and they were like, what is your problem? and they're saying, yeah, come on down. So it came down. I had my daughter with me. She got to excavate a mammoth in a little pink dress when she was 4. So that was awesome. And when I was there rummaging around, looking at the site, I walked up the grade a little ways to, the mammoth was found here, up this grade, and it's hard to see, but there's a rise here. And this is a kind of a side cut, and they cut it back to expand this road to give it a better grade, and so on. And one of the things I noticed was shells. I went, wait a minute, there's shells, it's sandy, it's kind of that clean sand that I saw out at the Fairmead landfill before, and I went, the shells, and alarms started going off. So I scooped up some matrix, and I found some bones in the matrix. So I ran down and said, I want as many sandbags as you can. I filled up a bunch of sandbags. And then next day, a week later, we drove up and excavated a 1x1 unit through this layer, which is stratigraphically below the muddy silt layer that the Mammoth lies in. So again, this is right along the San Andreas Fault in the Pajaro Salinas River system, and a lot of these low sand hills where they grow amazing artichokes that you can get at the Big Artichoke. Are, you know, they're folded and formed, but there's a really good stratigraphic sequence, I hope somebody drew it, of this wall. And it turns out that I was able to date an oyster shell to 29,900, which is, you know, clearly stratigraphic, a different stratigraphic unit below the Mammoth, and that made Mark sad, because he's like, oh man, that's too old. It's a different stratigraphic unit. The Mammoth could be 14,000 years old. We don't know yet. So actually, I have a chunk of bone being run by Brian Damiata at the Keck Lab at Irvine using new techniques that might be able to get some collagen out of this mammoth bone, so might be able to directly date the mammoth. And I'm hoping that it comes back 14,000 with hair, and I'll convince Mark to write up an NSF grant and end up in National Geographic, or not. Who knows? But the thing that really got me excited was the matrix and the diversity of these materials. And what did I do once I extracted fish bones? I sent them to Cango Ballet, who identified 3 freshwater fish in what looks like primarily an estuarine assemblage that probably contains wash-in material, you know, high energy wash-in that hit some cline, and then it became low energy, and all these bones and shells and things just deposited right there. But there's a tule perch, thick-tailed chub, and Sacramento sucker here. And these are all fish that Ken has identified earlier in the Pajaro Salinas drainage from archaeological sites as well. There are a lot of salamanders here too. All of these are represented. Aeneades niger, Aeneades lugubris, Batrachoceps, and Encetina schultzi. And I, you know, playing with Mapper, I decided to look at the collected range of Aeneades niger, and then drop in this 29,000-year-old record for Aeneades niger, which is in a flat, at well, close to the nick point, but in these rolling hills, fairly well out of what is normally considered the forested preferred habitat of Aeneades niger. The caveat is that these bones are probably washed in from higher up. And these are data tables. Again, the fish material identified by Cango Ballet includes marine, euryhaline, and freshwater fish. Most of the shells are small oysters, clams, mud drilling clams, and other things that are upper estuary. There are also a wealth of ostracods, forams, as well, and I'd love to find someone who'd like to look at ostracods, because I've got great samples that are dated, and there's a diverse array of ostracods available. So if anybody has ostracod pals. He put us in touch. I feel confident that I was able to differentiate Anides niger, from lugubrious because I found caudal vertebrae of both and compared directly to both species and found that the caudal vertebrae of Anides niger, not having a prehensile tail, do not have all the flaring bone and muscle attachments that you would expect to see in Anides lugubrious, readily prehensile tail. And that's what I based the differentiation on. And you know, there are plenty of herps, plenty of mammals, and one of the things I want to do is go back to the site and get more larger bulk samples, because this is from about a cubic meter or maybe a meter and a half of sediment. So again, the site is very rich, and it's still there. So now I'd like to switch into the Holocene and the archaeological realm and talk about herps in particular from the Emeryville Shell Mound. How many of you know where the Emeryville Shell Mound is? Good, quite a few. It's under the IKEA store. That's where it is now. And the Emeryville Shell Mound is one of the most famous archaeological sites in the western U.S., without a doubt, if not the world. This is what the shell mound looked like in 1903. That large heap was leveled, and a covered dance floor with a bar was put on top of it for people to come dance by the bay. And of course, all of this is all archaeological too. But this is a very impressive shell heap. It was, what, at least 30 feet high, if not higher, maybe 50 feet at its peak, before its peak got chopped off. But this shell mound accreted over several thousand years and contained not only habitation debris, but also graves, burials of literally hundreds of Ohlone people. Some Kent Lightfoot's research has shown or illustrated that these coastal shell mounds were certainly central places, places of reference, places of where you could anchor your identity, where your ancestors are buried, and are, you know, very important features on the landscape, filled with archaeological data. And this is right by Temescal Creek. It must have been a wonderful place to live. You know, bayfront, 1,000 years ago. I mean, I'm not going to wax romantic. It would have been difficult, but there were certainly a lot of resources at the time. An unpolluted bay filled with sturgeon and salmon, sea otters. Yeah. Emeryville Shell Mound is also known for being the first real stratigraphic archaeological excavation in North America, if I'm not wrong. Although I guess Thomas Jefferson drew lines on some of his maps, but you know. No, Max Eula dug a big hole in 1903 and actually drew these lines in, drew the stratigraphic, and kept that data and paid attention to the artifacts as they related to the stratigraphy in the site, and was really the father of modern stratigraphic-oriented archaeology in North America, having studied Jens Warsaw in Europe earlier. And this is what ultimately happened to the Emeryville Shell Mound in 1924. It was steam shoveled, leveled, and The soil was screened and sold as garden soil, which explains to me my conundrums as a high schooler doing yard work in the Berkeley Hills when I would find these bits of soil, patches in backyards that had shell speckles in them. It's like, what's going on here? But there was a cottage industry where people would come out and screen all the rocks and junk, and not that I mean it's junk, human remains, mortars, spearheads, pestles, artifacts, and buy, sell, and trade those, and then package up this fantastic soil for yard soil. And you can thank Fuller O'Brien for that. And since they put a paint factory on it, the material I got to analyze, which was excavated in 1999, was excavated by crews wearing full hazmat suits with in 2,000 respirators. They were all HAZWOPER trained because there's cobalt and lead and acid and all this stuff in that soil. And even with the leveled mound, I think close to 200 human interments were found and eventually reburied. In terms of the overall herp assemblage, and again, this is where I want to focus on not so much the archaeological aspects of the faunal remains, but the non-economically important animals, the small things, the local things that represent the local environment. There are 14 genera and 10 species of herps representing 9 families, not including 9 freshwater anadromous fish, 8 genera, 11 species or families, 2 salamanders, 3 anurans, 2 lizards, 5 snakes. And just to show this is an example of analysis of trends over time of economically important animals at the Emeryville Shell Mound. These are probably the 5 most common large sources of mammalian meat at the site, broken down into these time periods with bunny rabbits, harbor seals, canis, still unsure whether it's coyotes or dogs, black-tailed deer, and sea otters. And sea otters represent a large proportion early on of the mammal remains. And then there's this radical change around between 1,500 and 1,300 years ago that times closely. Jack Broughton and I disagree about 100 years or so, but I suspect this may hint at a change in hunting technology when arrows, sinew-back bows showed up as opposed to atlatl darts, which may have increased hunting efficiency for deer as opposed to sea otter, and you see deer take off. Jack thinks that people were accessing unused space in the East Bay, but this was at a time of demographic expansion, and I would venture to guess that there wasn't a whole lot of unused or unoccupied or unowned or uncontrolled space. So my argument is technological advance. But that's another talk, right? This is where the Emeryville Shell Mound is right now. Notice, Shell Mound Street. You can go along the railroad tracks here and still find shells if you look hard enough. I tried to cover it all up, but it's still there. Lots of people drive by that every day. Millions a day, probably. Now, Cango Ballet, again, identified, along with Kaylee Harden, identified the fish remains from the 1999 samples at the Emeryville Shell Mound. And, you know, it's bayfront, right? So there are going to be a lot of marine fish. There were. But there are also a lot of more sensitive anadromous fish and freshwater fish included in this assemblage. And it's important to remember how amazing San Francisco Bay must have been 1,000 years ago in terms of anadromous fish resources, migratory waterfowl and bird resources. There were elk here, there were pronghorn here, there were lots of deer, sea otters, arbor seals, a few sea lions, murre and cormorant rookeries on all the islands. There were a lot of sturgeon that were identified at Emeryville, and sturgeon are really, really nice. Their caviar is very nice. They're large, tasty fish that provide a lot of meat to an assemblage. Kate is responsible for these numbers, and they're fantastic numbers. 2 species of sturgeon represented, and then other anadromous fish, of course, include salmon, silver salmon, steelhead, and king salmon, again in large numbers. But there are also freshwater fish represented, which makes me wonder if these were caught in local area creeks or represent maybe a freshwater fish fauna for Temescal Creek, or Codornices Creek, or Strawberry Creek. And the freshwater fish identified include Sacramento perch, Sacramento pike minnow, Sacramento split-tail, thick-tailed chub, hitch, and Sacramento sucker. All in low numbers, but they're all represented. And these are what they look like. Split-tail, Sacramento perch, thick-tailed chub, hitch, Sacramento sucker, and Sacramento pike minnow. In terms of herps, these are the denizens, none of which I would particularly not expect to be here. And I wonder also about abandonment of the shell mound. Were people always on it, or were oak trees, could trees have grown over a period of time? It's hard to say. Maybe these creatures came in naturally or with firewood or what have you, but they're all present. Rana aurora, Ensatina, Schultzii, Xanthoptica, Bufo boreas, Netes lugubris, and Pseudacris are all present. And here's the numerical data slide. One thing I want to point out is that you can see the number of units. These are excavation units, some of which are a couple meters deep, that I identified and that these data came out of. And really, if they're these herpetological remains are rare in these archaeological assemblages. And at Emeryville, we only have the assemblage that we do because such a great volume of material was excavated from the site. So as a result of that huge sample, we're able to get at least kind of a hint of the herpetological diversity at Emeryville. I'd like to briefly stop just inland from Goleta Slough in downtown Santa Barbara, SBA 53, which is a business park project in the center of town in 2015. And the fine screening at this site produced 12 genera and 10 species of herps, representing 9 families. We have salamander, frog, 5 lizard, and 5 snakes. This is a location of the rough approximate location, SBA 53, up above Goleta Slough. And there are 2 salamanders actually. No, I didn't find any tracheoceps here, but Netes lugubris is represented, western pond turtles, Bufo boreas. These are the data numbers. 1 Netes, 5 Bufo, a few pond turtles, surprising number of lizards for this small sample size, and several snakes. we're almost done. I want to talk about two more sites. One, which I just had the pleasure of analyzing 2 years ago, which is, it was a sample from the site of Humaliwo, or Malibu, probably the largest chiefly village in the Chumash world at the eastern border of their territory. Basically, Malibu Creek and the Malibu Village site demarcate close to a fluid cultural boundary between Chumash and Tongva in the LA Basin. Now this site is right on Highway 1, the Coast Highway, Pacific Coast Highway. It was excavated when the highway was originally expanded in 1965, again in 1985, and then a part of the site was excavated in 2018 to maintain one of the historic structures, a lath house, on the site. And I would like to add that this is one of those large mounded coastal shell mounds, not as big as Emeryville, but you can actually see the rise when you're traveling along PCH and look oceanward of the highway. And the excavations in 1965 produced one currently protected species. The 1985 excavations produced two currently protected species, and the sample I analyzed from 2018 produced 7 currently protected species. And this doesn't have a whole lot to do with change in protection law. It really points up the differential recovery of this sample related to fine screen sizes. And that's one thing I can't emphasize enough. All of these samples that I was able to identify were screened through, at a bare minimum, 3 millimeter mesh, most of them through 1 millimeter mesh. The archaeological standard in California and throughout much of North America is 3 millimeter mesh, but now people are at least taking column samples to run through finer mesh screens to do flotation analysis and collect plant remains in a standardized fashion. But also in the heavy fraction, you find these small bones. And you know, I've heard this mantra, it's been beaten into my head since grad school days. In order to excavate an archaeological site, you destroy it. So what you do is irreplaceable. And I would like to try to promote more fine-grained analysis of these sediments that come out of the ground. But again, it's cost prohibitive in terms of budget, time, interest. You give me the budget, I'll make the time, I have the interest. But there's a lot of utility to doing this, especially now as extinction rates are climbing, we have climate change to deal with, and humans are expanding across the landscape. The Holocene fossil record, the late Pleistocene fossil record, offers a real window into microvertebrate distribution in California and everywhere. Now the site, this is Malibu Creek, Malibu Estuary. The site that this was excavated from here, this is Malibu Shell Mound. There's a house there that was being strengthened. So it comes from right here. One of the interesting things about Malibu Creek is that it's one of the most southerly, am I correct, steelhead runs, native steelhead runs in the state. At least, there are probably some in Orange County, maybe.
Well, we found, didn't you find bones in San Diego County?
Yeah, I found 4 trout
My
vertebrae in the San Diego River.
Yeah.
So, and Ken's been working on distribution and paleo distributions of salamanders in California for quite a while, which, as we know, are declining. It used to be much more prevalent in the state and an important food source. There's a dam right about here, that was placed in, put in 1925 or so, that basically stopped the steelhead run. There's historic references to great steelhead fishing in Malibu Creek, and there's photos and accounts of people going up to the dam to go steelhead fishing because it was great. The steelhead would concentrate there because they couldn't go any farther. And some still did breed in the lower reaches of the creek, but Ken looked at fish remains from up in Malibu Creek State Park, some sites there that had salamander or steelhead in them. And lo and behold, in going through these sediments, I found a few trout, Oncorhynchus, Micus probably, vertebrae, along with Rhododratenii, Actinemes marvarata, and Phrynosoma blainvillei, a couple diagnostic head bones. These are all protected species now, certainly, in Southern California. Another protected species I found in the assemblage is this guy, a longtime favorite of Dwight Simon's, short-tailed albatross. They're represented here, as are southern fur seals and California sea otters. And some of the park biologists in Malibu Creek State Park were enthused with some of these results in terms of, they can add these findings to some of their reports in terms of conservation and management programs, saying, yeah, we've got old records, archaeologically dated records of red-legged frogs, and even further support for steelhead in Malibu Creek. And one of the big projects that people are really pushing for now is to remove that dam because it's silted up completely and is, is worthless other than maybe as a mallard pond. And there's a real push to take that dam down and let the stream flow naturally with the hope that steelhead will come back. And steelhead are coming back into the lower reaches quite regularly. I'd like to finish up with a site, the Avenal Creek Bridge site that was part of the State Route 41 expansion and improvement project that was managed by Bob Reynolds, who might have come to a Herp group or two, I suppose. He was a paleontology student here. And this assemblage comes from Avenal Creek. It's right about here. This isn't high enough resolution, but this is about where the creek, the State Route 41 crosses over Avenal Creek, which drains this section of mountains. And this is Parkfield. So you see these from Highway 5 off in the distance to the west. This assemblage dates to 946 years ago from a direct radiocarbon date on charcoal, just prior to the medieval climatic anomaly in California. And the materials here include 28 species, 30 genera, and represent 18 families. Include 2 freshwater fish, a salamander, 2 anurids, 7 lizards, 7 snakes, 11 mammals, and 4 currently protected species. The fish from Avenal Creek include Sacramento perch and hitch. Not unexpected, but nice to know they're there. I haven't checked to see if there are records of these fish from Avenal Creek. I'm not sure, but we can do that. The amphibians include Anides lugubrious, which appears to be in almost every assemblage you look at in California. Spea hammondii and Bufo boreas. And I went to Mapper again, and I mapped collected range or known records of Aenetes lugubrius onto and expected range of Aenetes lugubrius, and the red triangle represents the Aenetes specimen from Avenel Creek, right at the edge of its expected range, but nowhere close to any known records. So I would argue that now you have a known record right at the edge of its expected range, substantiating that border. Other specimens we found or I identified include Gambelia sila, currently very, very protected, as well as Dipodomys ingens. These bones were Dipodomys, and they were giant, so I identified them as the giant kangaroo rat, bigger than any others. And then antelope ground squirrel as well. And these are numbers. There's some interesting things about this site that I'd like to wax on for a little bit, not too much. The herp diversity, especially the reptile diversity in this collection, is really high. And these are fluvially deposited sediments that probably hit a low energy area, swirled and settled. Just happened to be right where the bridge abutment was going to be. It included the two fish, the amphibians, but quite a few lizards, arid lizards, and snakes. Some less common snakes archaeologically, like Hypsiglena and Rhinochilus, but over 200 horned lizard bones. And also lots of pocket mice, rodents, and things. And one thing I noticed about this assemblage, a lot of the horned lizard bones, some of the snake bones, many of the rodent bones were charred or calcined or burned. And that kind of flipped a switch, and a dim light bulb flickered over my head. And I went, this really looks like something that could support an inference of a post-catastrophic wildfire rain washout event, where you have a wildfire that burns aggressively over the ground and kills everything that can't run faster than it. And interestingly enough, this past spring I led an archaeological survey class in the Santa Monica Mountains in the Woolsey burn zone, doing post-fire survey. And one thing I noticed, and I pointed out to all the students, were these burnt carcasses of wood rats, small mammals, rattlesnakes, gopher snakes. We found cooked Aniella, cooked Elgaria, and so on. And that really kind of rung this bell for me even harder. And so, finds like this are at least a data point or something that can be discussed in terms of California wildfire history and effects on wildlife that aren't often touched on because people don't often look at the microfauna as opposed to the macrofauna. And I want to emphasize that none of these assemblages would have been collected were it not for the California Environmental Quality Act, California Public Resources Code, and NEPA, National Historic Preservation Act, Section 106, 36 CFR 800, all these environmental laws that were enacted partly by Tricky Dick and are under assault now. And with that, I'd like to thank you for your kind attention.
Let's take a few questions.
Yeah, Tom, since it's 50 years ago, Tandy, I guess.
Yeah, it's fine.
When you dismiss a lot of these herps as not important or non-culturally important. Now, when you get an unusual site like the one with many Tarika that you never find before,
Yeah.
can you think of they might have been used for something?
Well, there's an important distinction here, and that has to do with dating and time of those sites. The Pleistocene sites that I looked at, I'm convinced are not archaeological. So they're Pleistocene fossil record, naturally deposited fossil record, like the site I just talked about, in the sense of a naturally occurring fluvial fossil deposit. But notwithstanding, archaeologists, zooarchaeologists do often dismiss animals that are not the important meat animals as not economically significant. And I mentioned at the beginning, there is growing evidence for use of non-economically significant animals in terms of ritual, healing, maybe medicine, that is just not very considered by zooarchaeologists. There have been a few studies on use of turtles as turtle shells as rattles that have come out recently. That certainly there are turtle shell rattles in the ethnographic record in California, so they could represent that, but it's a bit of a leap without, from my point of view, the positive evidence that would support that. Yes?
So you said that there were Aniella that were burnt up or charred?
Yeah, we found two dead Aniella in Leo Carrillo State Park that were on a sear, just burnt gravelly soil.
Could they just have been killed in a fire, or?
Yeah, I think they were killed by the
They
fire.
weren't like ceremonially seared.
No, these were on the surface. Yeah, these were on the surface.
Sure, Dad could
For-
come up with something.
Used as pencils afterwards or something.
Yeah. Yes.
So Tom, with the archaeological sites, and looking at the microfauna, which is really great, and the salamanders, et cetera, could some of those be part of the actual harvesting of other species that they're going after, with nets and other things like that, and that they're being brought back into the sites?
Discarded.
They're kind of part of that whole process?
They could represent bycatch.
Yeah.
It wouldn't surprise me if red-legged frogs were consumed, because, frogs are yummy. Now, and there's other things. When you think about ethnographic record of medicine and ritual in California, there are various kinds of poisoning. There are various kinds of medicinal treatments. And the people who lived here for thousands of years, I'm sure at least some of them were acquainted with the behaviors of some of these animals. Toads, Encetina and Tarika are both, Tarika especially, very toxic and can kill people. Encetina, I know people who've seen it in the field, but they exude a sticky, Elmer's glue-like toxic substance that is known to seal the jaws of a predator, Thamnophis, the garter snake, closed, so they can't open them. And there's also a poison. So it wouldn't surprise me if some of these animals could have been used in some kind of ritual medicinal way. The ethnographic evidence is scant. But if you look at other groups and their use of these kinds of animals, it does occur. Yes?
Hey Tom, I really like the story of your daughter and excavating the mammoth. And I was just thinking about that, was wondering if there have been any efforts or going on to bridge the work that you're doing in zooarchaeology public, or even with Tonga and Olney, like community groups and things like that, or with club educators?
There's not enough that goes on, but Kent in particular has been working a lot with local indigenous California groups. I try when I can. I work with some Chumash and some Tonga people, and since I do some consulting for them, and I'm constantly trying to reach out to people in terms of, you know, offering this kind of, isn't this cool, kind of casual approach. Look at these weird things. Who would have thunk that? And I found that to be somewhat effective. UCLA, we have an open house every year, and I also present to school groups sometimes and give public presentations around Southern California. And I hope my enthusiasm is infectious.
Thanks.
Yes?
What can your work kind of tell us about how, because I imagine that the animals that you found way back in the past are not necessarily the ones that you would find in those same spots today.
Yep.
What kind of information can that provide us for how climate change is affected, or just how the environment has changed in general?
That's something that really interests me, is especially the Pleistocene material, is looking at, you know, for example, chipmunks at Huntington Beach. Why? And I would point to climate perturbations and a different climatic regime at that time. But by getting more data, especially on small animals that are affected differently by climate than large animals, we can get a finer grained understanding of past climate fluctuations in California and what that might mean for past distribution of animals and future distribution of animals. And one particularly important theme is the medieval climatic anomaly in California, where around, 1300 AD, 1200 AD, there were severe droughts, severe climatic perturbation, such that many Sierran lakes have 10 to 20 feet underwater, groves of stumps that represent 100-year-old trees, which is indicative of severe drought and radical decline in water availability, and there are other inferences you can make from that, maybe temperature, exposure, what have you. But there is data available, if we look for it, concerning our most recent mega drought that can certainly inform us for the coming mega droughts.
All right, thank you very much, Tom, and thank you all for being here tonight to help us celebrate the 50th anniversary of Herp Group. And I hope I'll see many of you again in the future.
The appropriate. I'm going to say, as an outsider to this group, since I study things with fur on them, the thanks should go to you and Marvalee for starting this.
Yeah.
Here, here.
Yay.